right, should be up on YouTube in a second.
Yeah, I see it there.
All right, we're live up on YouTube in a second.
Yeah, I see it there.
Okay, so I'm just going to ask everyone to mute themselves except for Naama. All right. Excellent, then we're ready to get started. So welcome everyone at our second KNAW Biophysics Talks meeting. We're having these meetings now pretty much on a monthly basis, every second Tuesday of the month. And we start out the first hour with short talks focused on big questions and open challenges in the field. And in the second hour, we have more of a tutorial component with a much more interactive talk. So before we get started, I want to very quickly introduce our co-organizers. So I'd like to introduce Greg Stevens, my colleague from the FU, Liedewijn Laan from TU Delft, who couldn't join us today, but she's definitely on the organizers board, and we have Tom Shimizu with us today from AMOF. In addition, we also invited a few colleagues to join us on the panel, so they'll be participating in the discussion. I want to introduce Rutger Hermsen, who's an assistant professor in Utrecht. I'd like to introduce Maaike Hansen, assistant professor in Nijmegen, and Louise Jaworth, who is busy transitioning from her postdoc at the MPI in Dresden and will soon start as an assistant professor in Leiden. So welcome everyone, and thanks for joining. We're going to start with two talks, short talks, and we really invite you to drop your questions in the Zoom chat or the YouTube chat, and we'll read them out to our speakers during the question and answer session. Before I introduce the speakers, I just want to also quickly announce the next meeting we're going to have on December 8th, where we'll have Leonid Mirny and Agnes Seminara. So mark the date in your calendar, and I hope you also all join for that meeting. All right, so we're going to get started today with our first speaker, Naama Brenner. Naama is a professor at the Technion in Haifa, and her group studies cell and population biophysics. She started her career off mostly working in neuroscience, but then branched out in many different areas of biophysics with, I think, the role of variability and statistical effects in biological systems being a very major theme in most of her research. And this is also very much going to be a theme of her talk today. She's going to tell us about the dynamic basis of phenotypic variability. So Naama, thanks for joining us, and please go ahead.
Thank you, Chase, and thanks for the organizers for inviting me to this nice event. And I'll tell you indeed about phenotypic variability, and in particular its relation to dynamics in cell populations. So we all know that biological cells are different from one another. Each one of them is unique, and their variability is apparent in any property that we can measure, whether it's size, protein content, shape, anything that's measurable, we find variability. And my story today will begin with our attempt to quantify this variability in microbial populations, in particular protein distributions. So can you see my cursor? Can you see my pointer when I'm pointing?
Yes, we can.
Okay, so I'm pointing now to a bunch of yeast cells. These are budding yeast. You can see mother and daughter pairs that are expressing green fluorescent protein from metabolic promoter of the GAL system. And these cells are grown in conditions where this is a required enzyme. So we're not talking about small number fluctuations here. We're talking about a highly expressed protein, and variability can be seen even by eye. We quantify this by the distribution. We have fluorescence on the x-axis in arbitrary units, and we plot the distribution or the probability density in the y-axis. And you might naively think that for a highly expressed protein that's really biologically functional and required, you'll see a small Gaussian around a high value, but that's obviously not the case. What we're seeing here is a very broad distribution. This is taken by fluorescent microscopy. There are about 3,000 cells here. You can see that the standard deviation is on the order of the mean, and there's an enormous difference between individuals in the population. A more efficient way of doing these measurements is not by microscopy, but by flow cytometry, where you can get several hundred thousands of cells easily. But the picture, of course, remains the same. We have a very broad distribution. For comparison, this is what a Gaussian would look like with the same mean and variance. So this is a highly non-Gaussian distribution with a very long tail. And the measurement is very high quality. You can see that the distribution is smooth over many decades of probability. And so what we wanted to do was to really understand where this variability comes from and how we can characterize it. So what we set out to do in the beginning is to compare, to try to find, because we know so much about protein production in cells, our idea was to have a set of control parameters that affects protein production and to see how that turns out to affect the distribution. So we added to our budding yeast that we saw before, we added for a comparison E. coli cells, and we looked at analogous metabolic enzymes in the E. coli bacterium, but also other proteins. And we started to play around with these handles with control parameters of the population. For example, the copy number of the plasmid that expresses the protein. For example, growth conditions, whether it's batch or continuous culture. And we had a whole set of control parameters that we thought might affect how protein is produced. And we thought we might trace down what is a meaningful process that gives rise to these distributions. So as you can see, really changing parameters, for example, copy number obviously affects, this is high copy number and this is low copy number, affects the protein production, and it increases the mean, but with it, it increases the variance. And what we saw is that something is kept quite robust as we change parameters, and that has to do with the shape of these distributions. They scale out, they can become narrower or broader, but they're quite similar to one another throughout the entire set of control parameters. And to see this similarity better, what we did was we scaled the variables. So we scaled the fluorescence by subtracting its average and dividing by the standard deviation. And what we found was that all the data from all experiments collapsed on this one universal distribution shape, which is highly non-Gaussian, as we already saw. But practically everything that we could put into this parameter, set of parameters, could affect the variance and mean, but not the shape of the distribution. So when we do such a scaling to subtract the mean and divide by the standard deviation, we throw away, of course, information. And so you can bring back that information if you take for every experiment the mean and the standard, the mean and the variance, and plot them on the same plot. And perhaps surprisingly, even those are not separately containing new information, because the mean and variance are highly constrained to one another. They all lie for the whole set of experiments on this quadratic curve that describes the variance as a quadratic function of the mean. So basically everything can be inflated back to physical units, reading from this relation with one parameter. And there is a lot of, these distributions are very universal. And what this means to us, how we interpreted this, was that a lot of the microscopic detail that we know affect protein production is somehow buffered as you ascend to the population level of organization. And in particular, no one dominant mechanism can be said to give rise to this distribution. There's a whole set of mechanisms interplaying, and there's some kind of buffering, and protein content, it seems, behaves like a variable that is involved in a lot of processes at the same time that are integrated and somehow you become blind to these processes as you go up to the distribution. So universality like this, the collapse of distributions, etc., is very cool, but in some way it's a dead end result, because we were not able to trace a lot of things that we thought we might understand about the underlying processes, and it seems like steady state distributions that are taken at a snapshot hold rather little information. And so we wanted to append these measurements by taking a different point of view on the population, and that is through time. And so we went on to measure practically the same quantity, but in single cells across time. And for this, we used what is now a very popular experimental method for bacteria, and that is the mother machine. This is a complementary measurement that tracks single cells a long time. And the way it works is that you have a microfluidic trap. Bacteria are grown in the trap. They start to divide. The youngest daughters are washed outside of the trap, but then you can follow the mother cell or the bottom cell or several of the bottom cells through an extended period of time and see the dynamics. Typically, you can get 100 divisions. If you're lucky, you can get even more. And you can measure the protein content through green fluorescence proteins. You can also measure other properties such as cell size, etc. So in order to compare the temporal traces to the population statistics, what we do is we collected from this time trace the values of protein content, and we plot them in a distribution, in a histogram similar to what we did for the population. And already you can see this familiar shape that we had before with slightly different scales. But when you scale all of the protein contents in different traces, you get the same distribution shape. And interestingly, for cell size, the exact same phenomenon occurs. You plot the values along time, and you get this skewed distribution which scales upon the 2-parameter scaling. And you can compare that back to the distribution you had for the population. There's, of course, the question of statistics because the traces don't have that much statistics. So you can pool them together. And you see that you can say that this is consistent. So these squares are the pooled traces from the mother machine. The blue is the population distribution that we saw in the beginning. And they really are consistent with one another to the accuracy of the measurement. And moreover, there is still this very strong relation between mean and variance. And so here the colors are the population snapshots. The squares are the one taken from the traces, and they really sit on the same parabolic curve. So it seems that the statistical properties of these traces match those of the populations, and it might be a good idea to actually look at these traces more closely in terms of how they contribute to the building of the probability distribution. So
Naama, can I ask a brief question?
Sure.
Thank you. So what's not completely clear to me is the fluorescence that you're measuring. Is that the integrated fluorescence of a cell?
Yes.
Or is it somehow normalized by volume?
No, it's not normalized.
Okay, so the variability that you see is
about the density later, but what I'm showing here is the total integrated.
Right. So the variability that you see includes just the size differences that you see. Yes. Okay. So yeah, thank you.
Okay, so actually this is cell size, and what you'll see in a couple of minutes is that really cell size and protein content are really related to one another, but not so trivially. They're not just multiples of one another. Okay, so this trace happens to be cell size, but protein behaves similarly. It accumulates throughout the cell cycle, approximately exponentially. This is a very good approximation. You can practically throw away the little wiggles inside the cell cycle. And so what we are tempted to do here, we're motivated to write a mapping, which relates this variable, whether it's cell size or protein, at the beginning of cycle N to the beginning of cycle N+1. And that mapping includes a growth factor or accumulated fold growth, which is e to the phi n, e to the alpha tau. There's this exponent, and there's the time that it took, and so it expanded over this fraction, this factor. And then to the beginning of the next cycle, you have the division fraction, Fn, which is close to half, but not exactly. So this is a very simple mapping that describes this trajectory very well, but it's also a stochastic process, a discrete stochastic process with the inherent timescale of the cell cycle. Because you can see here, for example, the exponential growth rates are variable. If you put them all together, they're all different. The times are different, the products are different, and everything is stochastic. And if you write this down logarithmically, you see that all these stochastic or these random variables somehow add up. And if you would think of taking them to be independent variables, then this is not a convergent process. There's no limiting distribution. This is a mathematical fact. You can see this empirically from the data in the following graph. So this is cell size again, in the blue, every dot is the cell size at the beginning of the cycle. And you can see that it is kept in check in a relatively narrow range. If I now, instead of the real trace, I just draw at random from the distribution of any one of these variables, I get fluctuations that are really huge. This is a logarithmic axis. You see that the cell size can inflate by 2 orders of magnitude and back. And so there's something that keeps this stochastic process constrained. And that is primarily a negative correlation between the fold expansion and the initial size. So cells that start out smaller inflate more and vice versa. And if you go back with this information to the mapping, you can see that it gets into the mapping in the form of a restraining potential, like a parabolic potential. And what you get in the end is a discrete Langevin equation. Okay, so you can see that there's this negative correlation acts as a restraining force. The noise acts as something that adds the entropic part, and in the end, there's an interplay between the two that gives you a stable stationary distribution for this variable. And under some assumptions,
Naama, Chase here, just a quick question about what you were just showing. So is this, what you're proposing here is a fairly phenomenological way of looking at it, right? So you have an idea of what has to constrain it.
Of course.
You're not yet proposing mechanistically.
Oh, no, thank you. That's a good question. This is a phenomenological model, a mesoscopic scale model. I don't think I'm going to get to the end of what I had planned in the allotted time, but I think I can just say that doing this phenomenology in the end helps you to put some constraints on mechanism as well. But at this point, I'm saying nothing about mechanism.
All right.
Okay, will you let me know when I have like a couple of minutes left or something like that?
Yeah, you have about 5 minutes.
5 minutes, okay, great. So to cut a long story short, these two opposing forces that are the restraining force and the noise somehow balance to give us a distribution. And when you take these, when you evaluate these or estimate them from the trajectories, you don't have any more fitting parameters, and you can really reconstruct the distribution to high accuracy. And you find that there's this shape factor. So the log normal distribution, if you go back now to real values and not logarithmic, you find that the shape is described by a log normal. And the log normal has a shape function. So it doesn't have a fixed shape. All of these are log normal distributions with different shape parameters. They can be very close to Gaussian, they can be intermediate, and they can be really highly skewed depending on this shape. And the question is, what keeps all of our data in the same shape? We had universal distributions that collapsed one on top of the other. The log normal distribution only collapses within classes of the same shape parameter. And to realize what it is that's keeping, we plot all the data points. So we evaluate for each of the traces this restraining force factor and the noise factor, and we put them on top of a map that shows the contours of the shape factor. And so size measurements are in blue and protein measurements are in red. And what you can see is that the restraining force is actually quite variable across different traces, going from 0.2 to 0.4, all the way almost until 0.6, but they are spread in such a way that they are almost on a single contour of the shape. And that is what keeps the robustness of this distribution shape. And actually, if you take the size and the protein content, one on top of the other, you can scale them to be practically the same distribution. Okay, and this gives us, lets us think back to the question about the relation between protein and cell size. Maybe we should try to integrate all of this information about cell size and protein content into one unified picture. And here's just a collection of all the statistical properties that some of them I talked about. This is the universal distribution. Oops, sorry. These are the exponential traces. Actually, they're not only similar to one another, the cycle by cycle exponential rates are highly correlated. This is cell size exponential rate on the x-axis and protein exponential rate. They're one by one correlated very highly. And the restraining force that we saw before is a property both of protein and of cell size. And they spread out, like we saw that the different traces behave differently. They spread out, the slope spread out in a way that is typical to both of them. And just to say a couple of words about how we should understand the multidimensional character of this of the cell. And so people have addressed the relation between cell size or cell volume and protein. For example, it was suggested that ribosomal protein underlie this exponential or autocatalytic growth and cell size falls. And actually, it's also the opposite has been proposed as well, that volume is the intrinsic thing that grows exponentially, and proteins in general relate or depend on the volume. And the point I want to make is that when you go from a mean field or average description like this one to a model of what we have, which is single cells and single time traces that have to exponentially grow and divide, a passive coupling of this type cannot hold together a system stably, because if I have one variable that controls division through some kind of control mechanism, and the other guys, the other variables have the same, even the same exponential growth rates, that's not going to help. You cannot control a system of many variables just by having their exponential growth rates the same, because again, noise will kill you. Fluctuations around these exponential increases and divisions will make the other variables diverge, sorry. And so this is unstable to fluctuations. And the way we solve this is we went back to a dynamic. I'm going to skip. Yeah, I'm going to summarize. I'm just going to say that.
Yeah.
Right. Okay, so the bottom line here is that in order to describe such a system stably, you must have real interactions, dynamic interactions, and then you can really get back all the statistical properties of the experiments. So here's the summary. Yeah, we have two different kinds of ensembles. We have a lot of statistical properties that we can learn from. And in the end, I think, even though our models are phenomenological and our descriptions are mesoscopic, we can learn something about mechanisms by doing this. And let me just, okay, first let me say thank you to my collaborators everywhere. And then I can leave on the open questions slide while you're asking questions, maybe, okay?
All right, thanks so much, amazing talk. So just for everyone who's tuned in, please drop your questions into the Zoom and YouTube chats, and we'll read them out loud. I'm just looking at the panel, whether there are questions at this point.
I have a question, if I can start off.
Yeah.
So Naama, that was a great talk. If we step back and think about, living versus our own engineered systems, we don't usually engineer systems to have such variability, right? Our engineered systems do the same thing over and over again precisely. Why do we? I'm just curious why you think there's so much variability, and what's the functional role of this variability that you see?
honestly, you want my wildest answer?
Yeah, I love wild answers.
I think this variability, in the end, this and other types of variability make a biological cell into a learning system. And the difference between the biological cell and the one that we engineer is that we engineer it to do one thing. And it does that maybe very well, robustly, etc. But a biological cell knows how to develop novel functionality, right? And evolve and stand against unforeseen challenges and stresses and create novel phenotypes. And I think that biological variability in its many different aspects is something that supports this behavior. But you know, this is I can't say I have proof for that. I have evidence maybe supporting this picture. But I think you're absolutely right. You know, everything that we know from engineered systems is not really applicable here. It's a sloppy system in some sense. It has a lot of things that it doesn't care about at all. Mm-hmm. And it holds loads of variability in dimensions that are not important to it. But when the time comes, these dimensions might turn useful for something new or for something stressful.
Great, thank you. Can I also read a question to the panelists, Chase? So this is from Lawrence. The fluorescence data, was that all from plasmid expressed proteins?
No, it was so that we also had as one of the control parameters, we had plasmid versus chromosome based, and we had low copy and high copy plasmids, and both of them were there.
Great, thank you.
So I was also interested by this comment you noted about, you know, universality being a dead end to some extent. One thing I wondered about is, like, are there any indications of systems that you saw, or maybe even you can imagine engineered systems, like where you somehow using tools from synthetic biology, for instance, where you could actually imagine breaking down this universality? So are there any signals that would hint at that?
You know, we were actually looking, one of the things we were looking for is to see this breakdown. And my collaborator, Aris Brown, continued doing similar experiments in yeast and other experiments where he also looked at transients. And it seems like this is really a really strong constraint. And also in other, we looked at published data and data throughout genome-wide experiments, and it's very robust. It's something, yeah, I mean, I plan to say something about this in the other talk, in the next talk, because I think there's still a very largely open question there. Is about this universality. I think
we'll stay. Yeah. Okay. We'll stay tuned for that. I think Micah also had a question from the panel.
Yeah, thanks a lot. That was a really wonderful talk. I was actually wondering, it's a little bit along the same lines. I was wondering, so overall, there's this very strong correlation, right, between your size and the protein content. And I was wondering if there's any parameters or things that you can modulate, that you can think of, that would maybe break that correlation, or if you remove the correlation, can you see additional noise?
Right, so I'm looking for the graph of the density. I think that would be useful to look at, but I'm not sure I have it here. So the surprising thing is that although there is such strong correlation, here it is. Okay, let me put this on. Okay, this is measurements from an experiment where you have two fluorescent proteins in the same cell. Okay. And they're really highly correlated. Everything is really highly correlated. You might think that there's really not, it's a constant density. At some point, we were testing to see whether it's not just a constant density case or situation. And here's the distribution of the density. Okay, this is protein divided by cell size, and they're really broad as well. They're not narrow distribution. Can you see these distributions at the bottom? Yeah, these are density distributions. So I don't know how to, I don't know if that answers your questions, but.
So you think there's maybe like two levels of noise, one that's correlated with the size and one that's maybe independent?
Yeah, I think so.
Cool. Nice. Thank you.
So we have time for one more question.
I have a couple more comments from the question from our worldwide audience. Can I read a few?
Yep.
Okay, this is from Isabella Graff. I was wondering whether you have an idea where the homeostatic regulation comes from, in particular, why the parameters restoring force, standard deviation always lie on one contour of shape.
Yeah, that's a great question. And actually that also relates to the same question about universality, because, you know, although we kind of figured out, you know, an explanation, it's a kind of not a very elegant explanation. And it kind of has two layers. One of them is that in the region where the data lives, this shape parameter is not very sensitive to the thing that's varying. Okay, let me go back to this, maybe to this graph. Okay, so this is the parameter. Okay, this is the parameter, and it turns out that it's more sensitive to the noise than it is to the restraining force. But among different experiments, the thing that changes most is not the noise, but the restraining force. So it kind of, I don't know, it could be a coincidence, but this is what it is. The shape is really not very sensitive. The other thing is that among these values, 0.2, 0.4, 0.6, right? So between 0.4 and 0.5 and 0.45, the shape is very similar. It's not, you know, you need to go to extreme values of the shape parameter to see that this is really very different from this one. So again, this is a sort of an ad hoc explanation, and in some way, this is maybe the most burning question that I've ever left on the roadside, is that I don't know, I don't have a better answer of what holds this universal together. Sorry.
Thanks so much. So I see lots of questions popping up in the chat and in the panel, but we should move on to the next talk. But there's good news, is we're going to have round 2 with Naama in about half an hour. We can really get into the nuts and bolts of all this. So please, you know, hold on to these questions, and I'm sure we'll have more time to get back to them after Cliff's talk. So Naama, thanks so much for that. Big round of applause from around the room here. Let's see. So Cliff has already shared his screen. I'd just like to introduce him very briefly. So Cliff Brangwynne is a professor at Princeton. His group really studies very broadly principles of biological organization inside cells. And he's really been playing a pioneering and a leading role in this very exciting research field, which is all about membrane-less organelles and the role of liquid-liquid phase separation inside cells. He's been receiving lots of honors and awards, including HHMI and MacArthur fellowships. Final thing I would like to say is, you know, just based on personal experience, I know he's a very inspiring guy to work with. So I'm very glad that he's joining us today and going to tell us a little bit about the work coming out of this group at the moment. So Cliff, thanks for joining us. The floor is yours.
Okay, thanks, Chase. Everybody can hear me? Thumbs up? Yeah, good. Well, it's really nice to get to speak at this forum. I've chatted with Chase before, and he was reminding me about the KNAW building, and I went back to my files. I think I gave a talk at that building in 2005, and I was going to bring up some of the slides, but they're kind of boring. But anyways, it's although sitting in my house in New Jersey is not quite the same as being on the canals in Amsterdam, it's nice to have a virtual effect. I was on Google Street View and sort of getting myself in the mood a second ago. Anyways, thanks again for having me. Excited to tell you about some of the things we're doing in my group. So I'll set the stage here by saying that biological organization happens on many different length scales, and I would say we know of a fair amount about what happens on the smallest length scale, thinking about structural biology models and so forth. And we have a pretty good understanding of what's happening at the larger scales. You know, we think about vertex models for tissues or continuum mechanics kinds of models for, you know, these structures that form on larger length scales. But at the mesoscale, we understand, I think, surprisingly little about how structures form, you know, in terms of predictive, mechanistic, biophysical understanding. We really have basically no idea about how a lot of this works at that scale. So at that scale, this intermediate, you know, kind of micron length scale in cells, one of the first things that comes to mind are organelles, which are these compartments which give rise to you know, specific regions within the cell, specific compartments in which sets of biomolecules are localized, and that can facilitate reactions or sequestering and so forth. And usually we're thinking about things like the sorts of structures we studied in high school biology, like the endoplasmic reticulum or the Golgi apparatus mitochondria. That's really just the tip of the iceberg, as I like to say. There's a whole other class of organelles that are membraneless, so different than the kind of canonical vesicle-like structures that that we learn about. And so these dozens of different types of them, they're typically assembling both protein and nucleic acids, often RNA and protein. And despite having no membranes, they can maintain shape and be coherent structures over many hours. And for reasons I'll tell you about, these are increasingly referred to as intracellular or biomolecular condensates. And so the reason they're called condensates is because we now understand that in many cases, at least, it's clear that these kinds of membraneless structures assemble through liquid-liquid phase separation, wherein the biomolecules within a cell condense into these liquid-like states. And so there are now dozens and dozens of examples of these and more coming out every day. We had studied this with these germline P granules and become interested in nucleoli and many nuclear bodies. There's a lot in the cytoplasm, and many of the signaling clusters that mediate intracellular signaling are also seem to be phase-separated liquid assemblies. So there's a lot of excitement in this field. There's a lot of open questions, though. And some of it has to do with the molecular driving forces. And so I just tell you a little bit about that kind of as a way of background. Many of the proteins within these condensates that are important for their formation have what we refer to as intrinsically disordered regions. Some proteins are, you know, essentially entirely disordered, but in many cases there's a combination. So often they'll have an oligomerization domain, a disordered region somewhere. Often it's in the middle, or it could be one of the C or N terminus. And then often there's also a substrate binding domain. It could be an RNA binding domain, ubiquitin binding domain, DNA binding domain, et cetera. And so why don't I show you this? This is a simulation from our collaborators Rohit Papu and Alex Holhouse. And what it shows is a disordered protein. So, you know, we're all familiar with proteins as polymers, but often we think of them as little pebbles rather than polymers. But in many cases, they can undergo conformational heterogeneity and fluctuate around. And those polymeric regions are, one often views them as weakly sticky regions that can promote phase separation. And together with oligomerization, it essentially amplifies those weak interactions. This is if this doesn't make sense to you, it's fine, but this is a kind of really the classic simplest picture for phase separation, which is a Flory-Huggins model, or a variant of this regular solution model. And the idea is it's a free energy as a function of the concentrations, where you have a competition between entropy and interaction energies. And this 1 over n term is essentially the reason why oligomerization matters. It cuts out the entropic contribution such that those weak interactions can promote condensation to get you over that entropic barrier. Now, it's a little bit of background. I want to tell you some of the key questions, and then I'll go into a specific problem that we're working on. One of the key questions in the field is, what is the interplay between equilibrium and non-equilibrium driving forces? You know, we're we're often taught that equilibrium equals death, and so cells are out of equilibrium. And so to what extent can we use equilibrium thermodynamic ideas to understand it, or is that totally inapplicable? Second question is, can specificity be encoded with disordered proteins? So these kinds of IDRs, as I call them, have played a central role in a lot of development of the field. And there's a question is, can you encode specific biomolecular interactions with disorder? So we're used to thinking about, lock and key type model that's been dominant for many decades. But if a protein is conformationally heterogeneous, can you still get specificity? And a third question, these are related questions, but a third question would be, how do condensates contribute to biological function and dysfunction? So how are they actually promoting physiological processes, and how might that go awry and lead to diseases like cancer and neurodegenerative diseases? So those are some of the questions that are really central in the field and being tackled within the field. I'm going to tell you a story that touches on some of those questions, and I would like to give the acknowledgments now for the team that's been working on this. And first and foremost, it's a postdoc in my lab named Shunsuke Shimobayashi, who's really done the lion's share of this work. But we benefited from collaborations with another postdoc in my lab, David Sanders. And then on the theory side, Pierre Ranseret and Miko Hataya, all at Princeton. And so the question that we've been specifically asking, which is related to those three questions that I mentioned, is where and when do these condensates form inside of cells? So these are 4 different examples of native endogenous condensates that are forming under different conditions. It could be when the cell is stressed, or when there's damage to DNA, or when the nuclear envelope reforms and these structures reform. In any case, it's, I think, important and still poorly understood exactly what are the rules, predictive biophysical rules that say where these things form and when. And so this is the question that I'm going to tell you about some of the work we're doing to try to tackle. Now, if you ask that kind of question, where does one phase form within another? That's something that's ubiquitous in non-living matter and material science and soft matter physics. And there's a well-described framework called classical nucleation theory. And this is really a starting point for thinking about how this works. It's also has limits of applicability even within non-living matter, but we're going to use it as kind of a base launching point. And so if I think about something like the nucleation of a water droplet from the gaseous phase, what I need to do is form a cluster of these little water molecules to get the droplet started. But once I form a cluster, what you see is I've got an interface that's formed, and nature abhors a vacuum, but it also doesn't seem to like interfaces. And so this interface is an energetically costly thing, and so the system has to overcome that. And generally, the way this is described, I'm going to move the panel out of the way so I can see, is there's a free energy as a function of the size of the cluster that's formed. And so the idea is that the larger the cluster, then the greater the volumetric free energy, favorable change in free energy. If the system wants to phase separate, you could say it's thermodynamically favorable. But there's a price to pay, which is this quadratic dependence on the size, because it's an area, and then this gamma is a surface tension. And because you've got a competition between a squared and a cubed term, what that means is that there's a peak, a barrier that has to be overcome, or a critical nucleus that has to be overcome in order for the system to grow. So you're going to have fluctuations, and once the critical nucleus is reached, then it should be favorable for it to continue to grow. So this is kind of the base set of ideas, and we wanted to think about, is this useful for understanding things in cells, and by asking that question, maybe we can learn something about where and when condensates form, and again, related to the applicability of equilibrium kinds of concepts in cells. And so, so to do this, we've been asking questions both about native condensates and also using these biomimetic systems that my lab has developed, where we can actually control phase transitions in living cells using essentially mimics of what happens natively with oligomerization. And so the idea is we have light-controlled oligomerization of these disordered regions. So we shine a light on this system. It's based on ferritin, which is a 24-mer. And so depending on the ratios of the two components that we express in cells and the light activation levels, we can get different degrees of oligomerization of these disordered chains in cells. And we've done this for lots of different proteins. These are two that we often are looking at, called FUS, which is related to, it's involved in cancer and also neurodegenerative disease, ALS, and HNRMP-A1, similarly a key disordered protein. And this is a movie of this protein FUS, and you'll see that shining light on this cell nucleus, and you see that we get phase separation in this controlled way. The movie's on loop, so it just shows you again and again we're forming these droplets when we shine the light on the cell. And again, this is biomimetic. It's an engineered system, but we think it recapitulates the same combination of oligomerization and weak stickiness from the disordered regions that occurs in native condensates. Now, one of the nice things about this system is that you can start to quantitatively map phase diagrams in living cells. So this is an axis which is like a temperature axis, because we said, oligomerization is essentially modulating entropy, and just as we think about temperature in that way, this is the ratio of core to IDR. So when there's relatively sparsely decorated cores versus densely decorated cores, you have cells down here that do phase separate and cells up here that don't phase separate. We can also see things that make sense in the context of classical frameworks, in particular, what looks like nucleation and growth just inside this apparent binodal. And then if we go much more deeply supersaturated in the system as a function of this core concentration, we'll get what looks like spinodal decomposition. So here's a movie close to the binodal. This is, I think, the same one I just showed you. It's nucleation and growth. And here's close outside the spinodal region, you see that kind of characteristic morphology that we associate with spinodal decomposition. Once we pass this inflection point where the curvature of the free energy landscape changes, we're in the spinodal. So this is nice and maybe even surprising. We're inside of a living cell, and we can use the same sort of picture and the same framework to understand what's going on. Now, what's also interesting is that the nucleation rate, even if we're in the nucleation and growth regime, so not in spinodal, if we're close to the spinodal, but still apparently nucleation and growth, you get very quick formation, very quick nucleation, very quick growth. If you're instead just moderately supersaturated, very close to the binodal, then it takes a while for things to nucleate. You see nothing's happening so far, and then eventually we start to get droplets that form after some time, and there's only a small number of them. So there seems to be a dependence on the supersaturation for the system. And so we've been quantifying this by plotting the droplet number density as a function of time, and then just taking the derivative in the early part of the curve. So the nucleation rate we define as the rate of change of the number density of droplets that are forming as a function of time. And what's pretty cool, I think, is you can then you can color code these points within the phase diagram, and you can see essentially what I showed you qualitatively before, that the nucleation rate is, these points are all blue, the nucleation rate is relatively low when you're only moderately supersaturated, and it becomes, the nucleation rate becomes much higher as you get more and more deeply supersaturated. And we see that for a number of different IDRs that we've tested. In this case, I'm showing you both FUS and HNRMP-A1. The shapes of the phase diagrams are a little bit different because the energetics of interaction mediated by these different IDRs is different, but the same trend in terms of higher supersaturation means higher nucleation rate. So we can start to put this together and go a little bit deeper. If I quantify the degree of supersaturation, which we'll write as the logarithm of the total concentration divided by the dilute phase concentration. So this is like the saturation concentration outside of the droplets. There's some subtleties there we can talk about in the discussion, but this is how we quantify this, the supersaturation. If we then plot the nucleation rate as a function of the degree of supersaturation, we get these nice trends where, again, higher supersaturation means higher nucleation rate, but you see that it starts to turn over. The data apparently looks like it's turning over here. And again, both for these two different types of IDRs. What's interesting is we can fit this data to a functional form predicted by classical nucleation theory. So it's an exponential. There's a prefactor, which is kind of like an activation rate. It's related to also the number of nucleation sites. But then we can write this as a ratio of S star to S, where S is the supersaturation, and S star is like a threshold supersaturation to get to this turnover point. So it's essentially telling you when the nucleation rate starts to become independent of the degree of supersaturation. Now, within classical nucleation theory, S star is related to the surface tension. So it's telling you something about the surface tension, and how big that barrier is. We're going to be agnostic about its underlying physics, because in the cell, we don't know if we can really use this equilibrium interpretation quite strictly, but the functional form is well fit to the data. I think I'll skip this or just show real quick. We see the same kind of thing in the cytoplasm, same trends, and we can fit in the same way. One of the things you're probably already thinking about is that in structured environments, we know that this idea of homogeneous nucleation is really not applicable. There's structure which changes the interfacial energies involved. And you can see that if you think, if we have some seltzer water and look at a straw, you'll see that there's these gas droplets or gas bubbles are nucleating on the straw. And that's because the interfacial energy cost is decreased by having that happen. So you're essentially lowering this barrier. And so we wanted to explore that in cells. And so we've been using
Chase here, just a heads up that you're low on time.
I'm low on time. Okay, maybe I'll just show a couple more slides. I want to make sure there's time for questions. Thanks, Chase. Yeah, I'll show you a little bit of this just very quickly. What we do here is essentially we're tethering disordered chains to regions of the nucleus. And what that allows us to do is ask, can we localize nucleation to specific regions, much like we saw in the straw. And so what you find is that, in fact, we can nucleate by having favorable landing sites, but it only works if the degree of supersaturation is modest. If we're strongly supersaturated, it doesn't work at all. The second thing that I'd like to, and you can look at these nucleation rates as a function of supersaturation, and you see that when we have seeded nucleation sites, the nucleation rates are much higher than in the unseeded case. And so we can actually quantify that in this nucleation threshold. What's, I think, really interesting, and I want to emphasize this, is that the degree to which we can successfully nucleate in a heterogeneous fashion, in a controlled fashion, depends on the disordered region that we tether. So this is what I showed you before. If the seed is the same type of seed, so it's a FUS seed and we're nucleating FUS condensates, it works very well. If we have mutated FUS, where some of the aromatic residues are removed, then it no longer works, even at very modest supersaturation. And then if you have different types of IDRs, it also doesn't work. So you don't get seeding at those sites. So I think that's pretty cool. And that gets at this, this question of specificity mediated by disorder. So I'll just very quickly show you something that I think is interesting. When we do the same measurement with endogenous condensates, what we find is that the S star values, the nucleation thresholds, are very low. So in fact, we can only establish upper bounds on those. And we think what this means is that the cell has optimized how it's doing it. So, if you're only moderately supersaturated below, these specific nucleation thresholds, then you'll have very slow but specific nucleation. And so it's going to be slow though. On the other hand, if you are super highly supersaturated, you're going to have non-specific nucleation. And so we think a cell, and we even have a mathematical criterion for this, is in a regime where you have specific but fast nucleation. And we can discuss this afterwards, but in some ongoing work, we are examining the mechanical interplay and the mechanical impact on nucleation. So I think I mentioned the key folks in the lab already, so I won't go through everybody, but just want to thank people in my lab, and I'll list these questions which we can discuss in Q&A.
That's great, Cliff. Thanks so much. Very exciting work. So we're going to open it up for questions. I think there's a question from Greg. You're muted, Greg.
Sorry, this is a question from Case Decker. There is a bewildering variety of phase-separated condensates and nuclei. How are these mutually distinguished? What drives certain molecules to some condensates and not to others?
Yeah, Case, thanks for the question. Yeah, I mean, that's a good, it's a great question, and it's certainly, this question of specificity and how specificity is encoded. One of the examples that I think about a lot is in the nucleolus. So the nucleolus seems to be a multi-phase condensate where there's a core set of proteins that condense into a liquid, and they're immiscible with an outer layer, this granular component, the outer layer. So it's clear that proteins and certainly RNAs as well, and my colleague Amy Gladfelter at UNC works a lot on this with RNA, but it's pretty clear that biomolecules can form immiscible phases, and that's not surprising based on some of what's known in soft matter and chemical engineering and so forth. So we think that the bewildering number of condensates in the nucleus, as you say, are mutually immiscible with one another. And how that's encoded is still a question that's poorly understood. But I think we're starting to get some idea on that based on combination of the kinds of tools we've developed and model in vitro systems. And yeah, it's a good question.
Great. I have one more that I'd like to read off. This is from Case Storm.
Mm-hmm.
Beautiful talk. Thank you. In the Flory-Huggins model, the interaction parameter chi determines where the system is relative to the critical point. In polymer mixtures, it is generally inversely related to the temperature, which clearly isn't changing much in cells. What determines the value of chi inside the cell, and how, if at all, is it modulated at all to induce or suppress phase separation?
Well, first, let me say it's great to be at a Dutch meeting and have the first two questions come from my favorite two cases in the world, so that's great. But yeah, no, seriously, this is another good question. Thanks, Case. And yeah, so that Flory-Chi parameter in cells, we think about it being mediated. Let me say the way that people have primarily been thinking about this is mediated by patterning of the disordered chains. So it's not only what amino acids are there, but their relative positioning with respect to one another. And that's been emphasized, for example, with respect to charge. You have blocks of positive charge and blocks of negative charge. Some really nice work by Tim Knott and Andy Baldwin and Julie Foreman-Kay. Also, the patterning of these aromatic residues, how many of them, what their spacing is. And so I think through combinations of that and post-translational modifications where cells now have non-equilibrium knobs that they're turning through the kinase activity and phosphatase activity. So we think about this chi parameter being something that cells can tune. So yeah, I think that's probably one of the main reasons that's done. And certainly as well through what I call, kind of like an entropic knob. If you, for example, post-translationally modify the oligomerization domains and then they assemble and disassemble, now you're effectively modulating the entropic contribution.
I have a few more questions from the audience, if that's okay, Chase.
Could we just switch to the panel real quick, and then we'll switch back to the audience? So Micah first, and then Louise.
Thanks, Chase. I'll be quick. Really nice talk. Thanks a lot for that. So I was wondering about the half-life of these condensates. Do you think that's related to cell cycle? And how do you think that impacts the ultimate function of these condensates?
Yeah, no, this is also interesting. And, I guess it depends on what we mean by half-life. So one of the things that's remarkable about them is that they, if you just made a movie in the microscope, you might say, okay, they're just kind of sitting there, and in many cases their sizes aren't necessarily changing so much. But you do a simple FRAP experiment and ask about exchange, and what you find is that they're completely turning over, like completely new set of those molecules on time scales of as little as 10 seconds or something. So they're super dynamic in that sense. They have a very low half-life, but with respect to how long it takes until they dissolve completely, cells are definitely, as we talked about with post-translational modifications, getting things to assemble and disassemble, and in fact some nice work from Lucas Pelkmans at University of Zurich on how that's regulated throughout the cell cycle, these Dirk kinases and other things. So a lot of the nuclear condensates dissolve when the nuclear envelope breaks down. And so, so I think that's an interesting question, and there's lots of kind of nuances to it that are fun to think about.
Louise?
Great talk, Cliff. I was wondering about the surface tension, because it seems one of the things that's striking, at least as far as we know about protein condensates, is that they have really, really low surface tensions, right? Oftentimes. I was just wondering, were you able to estimate that, since that, I know it doesn't, it goes out in a parameter for many of the equations like the free energy and stuff like that. So
yeah, excellent. Yeah, no, excellent question. Thanks, Lola. Nice to see you. Yeah, and you're totally right. I mean, a lot of times people ask me about Oswald ripening and do we see that, and generally we don't see that so much. And I always think my standard answer is that the surface tensions are pretty low. You think about actually nice work from Lecker-Kircher on colloids, and great illustrative examples of how the surface tensions can be so low with macromolecules, because you think about something like KT over a molecular length scale squared, and these are macromolecular liquids. So it is challenging to measure. We've done it in a few different ways with different systems. One of the things that I'll say, just to keep my answer short here, is that if you look at how the nucleation rate changes as you approach the critical point, and I could show you some of this data, but what it looks like is that it's consistent with the idea that as we approach the critical point, the surface tension vanishes. So that that's an idea that people in condensed matter would that resonates as something that they know, and so it's interesting that it also applies here. So it does seem that there's something like a surface tension in a classical sense that is controlling. Even while I tried to, we've presented this S star in a kind of agnostic way, because I think it's definitely non-equilibrium features to it, but it is certainly surface tension is certainly involved in the threshold.
But you don't know what it is, like you don't see the interface fluctuate, for instance, right, in a significant way.
What the values are?
Like if it's.
Yeah, I mean, I can't give you a precise value for this system, but we know that it's micronewtons per meter or less. Okay. And all the measurements we've done, it's pretty low for sure. And cool. we have an active set of projects, trying to measure it very precisely and manipulate it and all that, but it is, it's hard.
Okay, then a second question, if I have time, just real quick. Just to clarify, so in the one case where you did the mutant FUS, where you changed all the aromatic residues, so you got phase separation, but you didn't have specificity. Is that right? or you did not have phase separation?
Yeah, I showed that very quickly. Let me explain. So what we were doing there was, and we've done this in different ways, and you know, I just kind of went through it very quickly. But what we were doing is saying, let's take this correlate fuss system, which, you know, nicely phase separates, and we can map the phase diagram, and we're going to use that same system, but we're going to try to seed those condensates by tethering different disordered regions onto a specific place in the cell, namely telomeres. Yeah. So if you tether FUS and then you try to, you know, nucleate FUS condensates, they nucleate on the FUS tether, the tethered FUS. And that makes sense. There's homotypic interactions and they form nicely there. So then the question is, well, if you start messing with the FUS that's tethered, does it still nucleate? And you can break it basically, right? If you mutate these aromatic residues, then you no longer get the unmutated FUS condensates to form on that site. Moreover, if you tether HNR and PA1, a different IDR, it also does not nucleate very well. So we can, you know, so there's definitely specificity. These are, I mean, no surprise to you for sure, but maybe other people, you know, they're just, you know, squiggly proteins that aren't functional or something. You know, there's some old guard that still views things that way. But there's clearly specificity in the disordered chains.
Greg, if there's still time for one or two more questions from the chat.
Okay, so first, Peter Ryan, I think we already addressed your question with Oswald ripening, so we'll go on to David Zwicker. How do you experimentally distinguish between the nucleation and growth regime and spinodal decomposition? Yeah, wow, this is all my favorite people here. This is great. So that is an excellent question. And, you know, I'm curious if you all agree with me. People send me emails afterwards with further comments, because this is a super interesting conversation. But yeah, it's challenging. And at this stage, I'm going to, you know, full disclosure, it's really just morphology. We're just saying, look, it's, you know, it really looks, you can do it a little more precisely with the, you know, as it's clear that you're approaching something like a spinodal for two reasons. One, that the nucleation, you know, the nucleation rates, you know, start to become independent in a, you know, completely flat of the supersaturation. But the other one is that, you know, the morphology does look very different. Now, I talk to my colleagues who do molecular dynamic simulations for a living on non-living systems that phase separate, and I say, what metric do you use in your simulations to say now it's spinodal versus, you know, mathematically we can say it's, you know, the change in curvature of the free energy, but in the simulations, and what they tell me is it's very difficult. So I think to myself, gee, if it's difficult for you, it's going to be difficult for me, you know, inside of a living cell with proteins and stuff. So that's, if anybody feels differently, you know, email us and tell us what to do. But we've played around, you know, taking Fourier transforms and looking at evolution of length scales and stuff, but it was never that clean. And then we were discouraged from those conversations with modelers. So yeah.
I think Tom had a question, and then we should probably wrap up.
Yeah. Well, first of all, thanks for a great talk. Yeah. And I was curious to ask, you know, more at the phenomenological level, yeah, how much structure there might be within these droplets. I mean, they're macromolecules, right? That make them up. So is there any orientational order that's still there?
Yeah.
And, you know, and yeah, I'm wondering if we should really just be thinking of them as liquid droplets, or if there's still some remnant order in there.
Yeah.
That could give it some, spatial.
Yeah, I mean, that's also a great question, and it touches on something that has been centrally discussed, and I would say even heated controversial ways in the field. There's people that think that you know, if protein's not folded, then it's not doing anything, and so there was an idea that they really are basically, you know, folding together, you know, forming dynamic amyloid-like structures in these droplets. But the data from the structural biologists, there's a guy named Nick Fauzi who does some really nice work on this at Brown. And there hasn't been, I would say, his data suggests that they're not necessarily doing anything different inside the condensate versus outside. There's definitely a lot of these structures that form where they're more solid-like, more viscoelastic, and there's a picture in the field where these structures are nucleating amyloid fibers that are pathological. So we're very interested in that. I think there's no clear answers, and it's even a little bit controversial. But certainly there's cases where there's going to be structure that forms of some type. Yeah.
Maybe just to add one last question: can you get a drop phase separation within the droplets?
Yes, and that's related to the first question I mentioned about the nucleolus. So the nucleolus is an example of that, and so I would check out. So we did a paper. Ferrick is the first author, 2016, in Cell. You could check that out because we showed you purify one set of nucleolar proteins, you purify another set. Each of them separately forms droplets, and you mix them together, and now you get droplets within droplets, which seems to recapitulate what the nucleolus is. And so and that's something that there's a lot of interest in that. Again, what are the biomolecular determinants of that immiscibility, and how does the cell harness it, and those sorts of questions. Very open and interesting.
Very cool. Okay, thanks a lot.
All right, thanks, Cliff. This was a fantastic discussion. Thanks for joining us today.
Yeah, thanks everybody.
Really appreciate it. So we're going to switch now to the final talk of today, but I would actually like to suggest we take a few minutes break. This will also give us some time to switch between presentations. So we'll resume again in let's say 5 minutes. And those of you, especially the students who have registered to be active members of the tutorial, stay tuned in because I'll slowly turn you into panelist mode so that you can actively participate. All right, so thanks again for 2 fantastic talks, and we'll see you again in 5 minutes for Naama's talk. All right. We can probably get Naama set up to get started on her talk. Naama, you want to start sharing your screen again?
Yes. Okay, can you see it now?
Yeah, we can see it. I think I also promoted everyone to panelists who signed up. If I forgot about anyone, that person can just write me in the chat, and I'll upgrade you to become a panelist as well. So the way we're going to do the second hour is going to be hopefully more interactive. So everyone who has panel privileges can just shout out their questions and comments during the talk, and we'll just see how this goes. And if other people have questions, they can still write it in the chat, and we'll try to keep an eye on that and read it out loud if possible. All right, Naama.
Okay, great. Thank you.
Maybe it's just good to encourage people to turn on their cameras as well. We want a classroom atmosphere.
Yeah, that would be great. Thanks, Tom.
Okay. So I was asked to present more informally like Chase said, the nuts and bolts and a little bit of the underlying, the backstage stuff. And there's a lot of things that one might talk about in statistical properties of biophysical ensembles. I chose two of them. 2 points. I don't know if we'll get to both of them, but at least I prepared two points to talk about. The first one has to do with universal distributions and buffering. And although I said, I said this sentence that universality is a dead end in some sense, I have to say that's a very painful sentence for me. And it took me a very long time to say it. And there's been a lot of thought behind that. And this is something that I wanted to share with you. And the second question is maybe a little bit more technical, but also might be interesting, is the question of the different ensembles in a population and how this relates to the concept of ergodicity in statistical mechanics, and in what ways the population is different from a statistical ensemble. So my starting point is this, a wonderful graph that we all were very happy about when it first, when we first got it. And as I said before, we understood this to mean that biological details are buffered and that you cannot reverse engineer this distribution to learn anything about the molecular details. And in many ways, this was reminiscent to us of what we know of the central limit theorem, right? Because in the central limit theorem, there's a buffering effect. And let me just remind everyone very briefly that the central limit theorem tells us that if you have a set of independent, identically distributed random variables, we're taking the vanilla version, right, the simple version with finite mean and variance, then the sum of these variables is going to be distributed in the limit of large n with the Gaussian distribution and with the mean and variance that we can compute and that scale with the system size or with the number of added variables. And if you start with any random shape of distribution, after actually not a very long time, you can immediately see the Gaussian. And it's customary to say that for the central limit theorem, if your distribution is not crazy, 30 is equal to infinity. So by the time you're up to 30 variables, you don't see a trace of the original distribution. And this is exactly the type of effect that we thought might be going on here, is that things are adding up in some way and integrating in some way that we are losing track of we're losing the possibility of tracing them back to the molecular scale. But of course, something has to be different here because we have non-Gaussian, highly non-Gaussian distributions. Let me just say one more thing before I go to the non-Gaussian is So the reason why the central limit is important is because we see Gaussians everywhere in real life, right? And so when we see a Gaussian, we think we understand that because we attribute that to a large set of integrated variables that are, at least to some approximation, independent or not strongly coupled at least, and if not identically distributed, then similarly distributed. And the classic example is given by diffusion, where if you start a set of particles, random walkers at a given point, it doesn't matter what they do for the random walk. It doesn't matter what kind of steps they take and what the distribution of steps is, or the distribution of times, at least in some cases. What you get after long enough time, you see a Gaussian distribution, and the Gaussian scales appropriately the way you expect it. And so this is the practical importance of the central limit theorem, besides it being a central theorem of mathematics, is that you see Gaussian everywhere in real life, and you think you understand them because of the central limit theorem. And so now we have this phenomenon that we have strong universality, but it is non-Gaussian. So now we start to scratch our heads and say, okay, what kind of limit theorem could this be arising from, right? Maybe there's a little bit different limit theorem. There are many limit theorems. Maybe there is one that describes this non-Gaussian. And for example, if you instead of adding up variables, if you take a product of variables, right? Then by taking the logarithm of this equation, you can see that the logarithm of x is a sum, and therefore the x will converge to a log normal distribution. But the point, the thing that makes this inappropriate for what we're seeing is the point of the shape of the distribution, because Gaussian distributions all have the same shape. If you scale them by subtracting mean and dividing by standard deviation, they all collapse on one another, right? We know that. However, the log normal distribution, that's not true. So if you have processes that are integrating through product, then your shape of the distribution is sensitive to the number of steps that you have in the process. Okay, so if you have, this is a graph that shows 10 steps together and 30 steps together. 10 steps added and 30 steps added have the same shape under scaling, but 10 variables multiplied as compared to 30 variables multiplies have different shapes. You're not going to get them to collapse under linear scaling. And so the symmetry properties or the scaling properties of log normals are inconsistent with this interpretation of what we're seeing as a multiplicative central limit theorem. So the next thing we thought about is there a central limit also? Is there a central limit theorem for strongly correlated variables? Maybe it's not the sum versus product issue, but maybe it's the correlations that are important. And if there is such a limit theorem for correlated variables, then there should be universality also of non-Gaussian distribution. We should be able to see these universal shape distributions in the real world. And perhaps surprisingly, we found that really empirically, people have seen a lot of non-Gaussian distributions and universal shapes. And let me just tell you like a couple of examples that we found. There's a huge literature on this. I think one of the first examples of this was given by, this is a paper by Brangwynne in 1998, a very short two-page Nature paper, where they claimed that turbulence has some relation or analogy to magnetization fluctuations in the XY model. And what they did is the following. They took experimental results from a previous paper. That's a really interesting experiment where you have two plates that are turning at an angular velocity, and in between them there's a fluid with a certain Reynolds number, and they have a feedback system that keeps the rotation rate constant, so that the Reynolds number is fixed, but then they record the power that they have to insert into the plates, and they get the power fluctuations inside. So it's an indirect way of measuring the power fluctuations inside a turbulent flow. And what they find is they can change the Reynolds number by changing the goal of this feedback system to the angular rotation rate, but and the distribution changes, but all of them have the same shape. And I'm not going to talk about this too much, but they're flipping somehow the axis. So these distributions are similar to the ones that we have, except they're flipped around the x-axis. But they still have one side which is very steep and the other which has an exponential-like tail. So all of them collapse, and then they did simulations on the XY models, and those collapsed. That's already also known. But the point of this short paper was that these two classes of seemingly unrelated distributions actually collapse on one another as well when scaled in two-parameter scaling. And so this has a practical application concerning the non-Gaussianity of the real world. You can easily find things that are 6 sigma away from the average, which in Gaussian distributions are much, much more rare. And this is something that, you know, in practical applications of turbulence might be important. But beyond that, the claim was that these systems have something in common, very fundamental in common, in that they have fluctuations on all scales. And in some way, this is reflected in the way they integrate the fluctuations over all scales is reflected in this universal distribution that they found. So this was, I mean, several works followed this. The same group of Brangwynne, Holdsworth, and Pinton eventually came up with the distribution formula under some constraints that is also related to extreme value limit theorems, and they went beyond equilibrium systems near a phase transition to also to these are equilibrium systems, other systems near a phase transition. This is total magnetization of Ising model, fluctuations of cluster size in the percolation model. They're removed from one another so that you can see them, but they actually all have completely collapse on one another. In the top, you have non-equilibrium models that are slowly driven by external forces and reach a quasi-steady state. And then you can plot the distribution of those fluctuations. And there is no theory. So there's been quite many years past since these works were published, but there is no theory for this strongly correlated systems. And in statistics and probability theory, there's actually quite a heated debate whether correlated variables at all exhibit any universal behavior, or whether correlated variables display distributions that are highly sensitive to details. So as far as I'm concerned, this is not only a mathematical open question, but it's also very relevant to biophysics, because if there is some universality class or some fundamental principles that tell us under what conditions many variables or many interacting variables integrate to give something universal, then I would say these conditions are very relevant to cell biology and to distributions that we see in cell populations. So that part remained open, and although we were able eventually to get some answer to our question, there is no theory that was satisfactory in explaining the universality of these distributions. And it feels like there is something there to be found, but we haven't been able to find it.
Naama, I think there's a question.
Yeah.
Go ahead, Lauren.
Yes.
Lawrence first. Yeah.
Yeah, I was just wondering, how can you distinguish if you see this distribution? Could it also be that there's some underlying physical deterministic system that shapes a part of the distribution that may now be attributed to noise only? Or how can you?
Right. So I'm not sure I understand what you mean by noise only. There are fluctuations. What do you mean noise only?
So for example, if you look at a pendulum, for example, I mean, a
Yes.
pendulum in the wind, right? It's a deterministic system, and then it has fluctuations.
Right.
And then if you look at the distribution of the positions, for example, then I'm not sure how to think
Okay,
about it.
so in some sense, this is what we were after in the beginning when we identified these control parameters, and we thought, okay, maybe we can say, here's the deterministic thing that shapes the distribution. We vary this control parameter, we have a knob, and when we change that, we're going to see how it affects the distribution. That was the original thinking. But I think the results have led us to think that there is something very buffered and very, it's an ill-defined problem to look at this distribution and try to find what is the underlying. And that's why, if that's the case, then there's got to be a reason why it's not reversible, and maybe that, this is a conjecture, but one of the mechanisms that builds up distributions that are not reversible to their details or to their underlying mechanisms is a limit theorem. So we were looking for a limit theorem. I don't know did I answer your question?
Yeah, I think so. Thank you.
Okay. Anybody else want to ask?
Yeah, I do have a question. So when you talk about correlated variables, so what exactly do you mean? I mean, I think I understand the words, but of course there are many models that produce correlated variables or time correlated variables, and we know that many of them, they could even result in Gaussians, or in exponential distributions. So you have to be thinking about very specific class of correlated variables.
Okay, so first of all, that's part of the question, but there is a part that is known, and that is the following. If you have a set of n random variables, okay, if the correlations are much smaller than n, and are over a scale which is much smaller than n, then you can do renormalization, right? You can divide your system into blocks of size n, and those blocks are going to be independent. And if you have enough of them, you're still in the regime. Yeah, you're still in the regime of the central limit theorem. Now, the turbulence system and the XY model near criticality, and a lot of these systems that these guys were talking about are characterized. This is what I meant when I said fluctuations on all scales. So the correlations extend over all scales up to the system size. And when you increase the system size, you still get correlations over the system size. So this is something that happens in physics. It only happens near a critical point, right? But in biology, actually, the experience tells us that oftentimes this is the case. There is no separation between the system size and the correlation scales. So you double the system, and then you get more correlations over the size. And so in some sense, there's this very long discussion about whether biological systems are near criticality. And I sometimes don't know how to understand this question, but in terms of correlations of statistical quantities, this is very well defined. If you have a system that is not thermodynamic in the sense that it's much larger than its correlations, then the system might behave like a system near a critical point.
But your model for the growth of cells doesn't have that type of behavior, or does it?
No, it doesn't. That's what I said. So my model doesn't have any of this. So that's what I meant. I mean, in some way, we have this is something we have not succeeded in doing. We have not succeeded in understanding whether or not there is a limit theorem that is relevant to these data. What we have succeeded in doing is constructing a kind of phenomenological model that starts from the dynamics and reproduces a lot of the things that we're seeing, which is fine. But my feeling has been for a very long time that there might be something deeper here that we still don't understand. We don't have that, right? So this is one of the questions that I was asked in the previous talk. Where does the universality come from in the dynamic model? It doesn't come from a limit theorem. It comes from a simple robustness of where the data happens to be sitting in parameter space, and the way that the robustness of the distribution is to its parameters. But it's not a fundamental thing of variables being integrated in a way that forgets the details. We don't have that. We don't have a model for that.
No, but I also would my intuition would be that the explanation for the system that you were describing in your talk.
Mm-hmm.
I mean, I think there the reality probably is that the system forgets its history in a certain time scale. So that explanation is probably more correct than
I think you might be right. And actually, if I go 2 slides ahead, we call this buffering by timescale separation. So in a way, in the end, this is the conclusion. This is what we could do with our data. We could say, okay, there's a buffering because there's a timescale separation. And the timescale separation is seen by the fact that I can throw away these little noise terms. You know, this is what I call noise. It might be even related to measurement noise or something like that. And if I throw these measurement noises away, or these small amplitude and high frequency fluctuations, I don't change anything about the distribution shape. The distribution shape is contained in all the long timescale or intermediate timescale information, right? So this is what we were able to do, and I think it's a good description of this particular system. But I'm raising this question because I think it's still an open question, and I think it might very well be relevant to biological systems. And it's a really interesting open question.
Thank you.
I hope I answered your question. Any more?
Naama, I had a quick question. Are you going to go back to that slide in 2 slides time? I'm guessing you're going to. Okay, then I'll get back to it.
Okay, I just want to finish the second. So the reason why we deal so much with scaling and with collapse of distributions is because tails of distributions are really hard to fit. So if you're looking for a theoretical explanation of your data, it's very difficult to distinguish between different long-tailed distributions, even if you have a lot of data. Anyone who has tried to do this knows that it's really hard to do. So we try to stick to these scaling properties. It's easier to stick to the scaling properties which are robust than to the actual fitting of something to a distribution. And the other thing that we use that I showed in the talk is the scaling between moments. So I would like to elaborate a little bit on the scaling between moments, because that also helped us ultimately to reach the conclusion about our system. So in general, Gaussian distributions do not exhibit a scaling between moments. You can take a collection of Gaussians. Their average and variance are independent variables. So all of these points are Gaussian. Every one of them is a Gaussian distribution. It has a mean and a variance. They're unrelated to one another. A scaling between variance and mean in Gaussian distribution only arises if you think of it in terms of the central limit theorem as the system size goes to infinity, because then both the mean and the standard deviation, both the mean and the variance scale with n, and the standard deviation scales with the square root of n, and so they are related to one another. So we try to look at these scaling relations, and let me just say one technical note. It's very customary in the field to look not at variance and standard deviation, but at some noise measures. And this, we found that it's really sometimes very misleading, because when you look at scaling between, for example, coefficient of variation, which is variance divided by mean square as a function of the mean, you're really looking at dependent variables. You're looking at two things that you measured separately, but now you're compounding them into a variable and something that's dependent. So if you plotted this coefficient of variation as a function of mean, you might be tempted to think that there's a scaling in this system, but really there isn't. This is the same data as here in this cloud, and you're only seeing this relation because you're not looking at independent measurements. You're looking at compounded things. So it's better to look at the variance. And here's an example of real scaling. If you have a Poisson process, then your standard deviation is proportional to the square root of the mean. If you have an exponential distribution, it's equal to the mean. And if you have a gamma distribution, it's just proportional. So these are really strong relations between the moments that can help you to detect what kind of behavior the distribution has. And going once more back to the log normal that I talked about before, the log normal with its shaped classes has the interesting property that within a shaped class, mean and variance are related to one another. But the parameter of this, the prefactor of this parabola depends on the shape factor. So it branches out to different parabolas. For each shape factor, you get a separate one. And then, and here comes, now it comes the part that was in the talk, how we figured out that really, for our case, it is the robustness of the shape with respect to the parameter. And once you understand that all of your system has the same shape parameter, then all the properties follow. You have both the collapse of the distribution and the scaling of the moment provided to you by the fact that everything is very robust with respect to shape. So there was a question about this again?
Yeah, I was just wondering about this, the buffering of the timescale separation. So in effect, what you're trying to say is, let's say you have high frequency, medium frequency, and low frequency fluctuations, then it's always, or your distribution is always defined by your lowest frequency fluctuations that you see.
Yeah, no, but here I think it's even better. We have, here the situation is even better because there is an intrinsic timescale. Usually, you know, how do you define what is low and what is medium? That's sometimes tricky to set them apart. But in this system where you have dividing cells, you have an intrinsic timescale. And what this buffering, why the buffering is so strong is because you can move from a description of continuous time to a description of discrete time. And all that matters are variables that are averaged over one cell cycle.
So in theory, if you did have a deterministic underlying mechanism like a pendulum, then that should be the most definitive, or if the time scale of that, let's say oscillation, then that should be the one that's defining your distribution.
Oh, well, there is a time scale, but that time scale is highly variable. It's not a deterministic system. I mean, it could be deterministic, but it's a variable system. It's not a time scale. So the distribution of generation times is quite broad. When you look at the distribution of these times TK for the K generation or the K cycle, they're quite broad in themselves.
Okay.
So what we did is actually to disregard that at this point, because we just jumped from cycle n to cycle n+1, and we didn't take into account this time. And the reason why this worked well, I sort of hid this under the rug, but the main reason why this worked so well is that the time and the growth rate are negatively correlated. So if you look at their product, you get something which is pretty independent across generations, and that helps you in describing things as a function of generation n+1, and really forgetting that there is a continuous time. But in a lot of systems, you can't do that. In that sense, the discreteness here is helpful to it's putting a really strict threshold on this buffering. More questions?
Yeah, I have a question. So
okay.
It might be a stupid question, but earlier you rejected the possibility that it's just a multiplicative central limit theorem at work. But now it seems like, if I understood correctly, things are being described by a log normal distribution here with the shape parameter. So yeah, maybe I missed why that.
No, they're described by a log normal, but that log normal is not a result of a central limit theorem. So what happens is when you write a discrete Langevin equation, you also have a restraining force. And when you try to compute the distribution, it comes out to be a log normal under certain assumptions, but it does not converge to a log normal universally, because the conditions of the central limit theorems are now not obeyed, because of the restraining force, your random variables you have a very strong decay of memory of what happened in the past, right? So you have to sum up a set of random variables whose distributions are changing too rapidly for the central limit theorem to be applicable. That technically, that's the reason. So the solution is a log normal to a specific special case that you can do under some assumptions, but it's not because of a central limit theorem that it's a log normal. It's a special case.
Okay, thank you.
So what we did is, empirically, we went and did simulations and saw that it's actually not that sensitive to the assumptions, right? It's similar to a log normal under many different microscopic noises, but still there's no theoretical reason why that should be so. It is so, but there's no mathematical basis for this that tells you that it's a limit theorem.
Okay.
Okay.
Thank you.
Yeah, you would probably have a lot more to say. So can I ask another question on this slide, or are you?
Oh, no.
If you prefer to go on, move on, I'm also fine with that.
No, I think it's better to keep the discussion going than to have more slides.
So I think I just don't fully understand yet why you say that this is because of a buffering of, but why it's because of a timescale separation. Because if I understand well, if the fluctuations on these short timescales would have a large amplitude and would actually be, they would actually contribute to the variance, but it seems to be that a large part of the variance is actually explained by the timing between the division events and the non-equal distribution of the sizes of the cells at the start.
Right.
And that to me seems to be independent of whether there's a timescale separation. More importantly, that the small fluctuations are just very small compared to, or am I misunderstanding this point?
No, I think maybe I should try to explain what I mean by timescale separation. Maybe it's because the amplitude is small, but the way that I think about it is that something in this data allows me to simply look only at timescales of a cycle and up.
Yeah.
Do you think that's only the amplitude? I don't know. It might also be true if the amplitude was larger. The main contributor to this distribution here, so the green is the one that actually takes the data points, and the black is the one that takes the approximation to the data points by exponentials. So this is what I mean by timescale separation. To me, the black, which it's just a collection of things that happen over timescales that are a cycle length or larger, right? It's actually a cycle length. That's a good question. I think this might have been also true if these fluctuations were larger.
So the way I think about it is, what I see when I look at this data, I see this a sort of smooth exponential growth and division event on a large scale, with added on top another noise. And if you write it like that, then the variance presumably is also the sum of those two variances, and the only way you end up with being able to throw away these shorter timescales is because their added variance is small.
Right. Yeah, that's right. But you're not even describing the actual measurement at every single time. You're only describing these effective parameters. But yeah, I see what you're saying. The reason is because these pieces of the cycles are so variable in themselves that their variance is much larger than this.
That's how I see it.
I agree. Yeah, I agree. In the end, it translates into the fact that I'm timescale separating. I'm throwing everything on timescales smaller than a cell cycle. But I agree that you can attribute that to actually the variance itself. Yeah, I agree. Okay. Okay, so this is eventually what we were able to do with our data. And I actually don't have that much more to say, but I would like to point your attention to the question of ergodicity that we've been now thinking about for a while, and just kind of to frame it. So we all know, the founding fathers of statistical mechanics invented this concept of an ensemble to help us understand the behavior of macroscopic system. And the idea is that macroscopic systems have so many configurations that instead of thinking of one copy of the system, you want to think of many, many copies. And in some sense, you want to think that what you're seeing is a reflection of this probability of the ensemble. But in order for that to work, what they needed to assume is the ergodic hypothesis, which means that if you leave a system alone in equilibrium, it will go through all of its states, or through its potential states, in a way that's compatible with an ensemble. So over time is the same as over many, many copies of the ensemble. And this comes kind of to illustrate this. If you have a probability distribution in phase space, it's higher in some areas than others, then the trajectory of the system is going to spend more time there. And this is a central kind of cornerstone of statistical mechanics. And if this is true, then we have justification to understand microscopic system through the concept of an ensemble. Okay, so we know also that they were not able to prove this, but we now think it's true for equilibrium systems, and there's a whole branch of mathematics now that's called ergodic theory, and it's actually exceedingly difficult to prove anything. Only in very, very special cases was it possible to prove things, but still this is the basis of statistical mechanics, and this is why it works. And when you try to carry this over to cell populations, of course, when we have single cell measurements and noise and everything, we want to bring everything in from statistical mechanics, but it's not that trivial, because a population of cells is really a dynamical system. And the cells each of them constantly grows, as we saw, and producing and degrading proteins, and it's once in a while it's dividing, and it has inheritance, protein inheritance, epigenetic inheritance along generations. So there's a lot going on, and it's not exactly clear how to carry over the concept of ergodicity and the concept of ensemble. And so what I would like to highlight is that there are several ensembles that you might I briefly alluded to this in the talk, but this is something that as people continue to analyze more single cell data, they realize that it's more and more subtle than once thought. And so I can, I talked about 3 different ensembles. One of them is the snapshot ensemble. You have a set of cells that you measure at a given time. Of course, this population encodes in it at the given time a lot of history and a lot of common ancestor, etc. But the ensemble is the fixed time ensemble. Oops, sorry. The other ensemble is the temporal ensemble, and here you really need to go into experimental effort to collect enough statistics to say something about, distributions, etc. And this you can do in time-lapse microscopy or in a mother machine. As far as I know, this is probably the longest trace that I've ever seen. It has 250 cycles of division and growth, and it's really stable. And the experiment gave us the statistics, and this enables you to treat this as not an infinite time, but a very long time statistics, and compare between the two, which is what we do. On top of that, most of the traces that you measure, is there a question?
Yeah, I was just asking, or wanted to ask a technical question for, is there a rule of thumb for how many generations you have to do for it to be representative, for the time trajectory to be representative?
So the point is, the funny thing is that if things were narrowly distributed, you wouldn't need that much time. But since they're very broadly distributed, right, and standard deviations are on the order of the mean or larger, then you need a lot of statistics to fill up the tail. right? Because if it was a Gaussian, then the tail wouldn't matter. It's not, it's very rare anyway. But for these kinds of distribution, like the log normal, there's a lot of information in the tail. And to fill up events in the tail, you need quite a lot. So I don't know. Most people work with, traces of several dozen generations. And then this brings me to the third ensemble, which is the pooled lineages. So because there's not enough statistics in 50 generations, what you might think of doing is just pooling them together, right? And making a population representative. This gives you time dependence, but you get more statistics by pooling together several lineages.
Can I ask real quick? So have you ever looked at something that are other than microbial cells? Like I was thinking perhaps in development where you have basically all these cell divisions that occur at exactly the same time. I don't know if you'd have enough generations to get enough information or something like that, because it bothers me, this thing that you can't really find something that doesn't fit. Like I want to try to get at the underlying molecular, what's going on. It'd be nice to know when you break this. So.
Yeah, I mean, I have not looked at this kind of cells during development. I mean, I would love to look at such data. It's really interesting. People have looked at mammalian cells. I think cell lines and different kinds of cells, mammalian cells. And also there's no synchrony. So in a dish, if you take mammalian cells, they have very broad distribution of division times. But yeah, it's a really interesting question, and I don't know. I haven't looked at these kinds of data. So is there anything more specific that I can answer then?
I was just curious because it's of course very easy to ask, oh, when does it break, without suggesting something. And so I was thinking, there in development where you have really where do you have a clock? There must be examples.
Absolutely, that's a great question. Right? I mean, look, I think all of this single cell analysis and all of the methods that that we're developing have been largely used in microbial population because these are the model systems that are easier to work with. But I think I would guess that a lot of this is going to be carried over to other experimental systems, once they are kind of constructed more in a very controlled way. And there's a lot of questions, of course, that you can similarly ask about, of course, about tissue and about development and about mammalian cells. Yeah, that's a very good question. But see, the thing is that even in bacteria, and what I would want to show you in the few minutes that I've left, I want to show you one instance where this separation to different ensembles, it really revealed something about the mechanism of cell division control. And again, I alluded to this in the talk briefly, but I'd like to kind of put it in context of these ensembles. So we talked about the restraining force of this stochastic process, the intermediate level, the kind of mesoscopic stochastic process. And these negative correlations can provide us with a good description of the limit of the distribution in these time traces. And if you look at the entire, what people do is they take a lot of traces, they pull them together, and they look at this correlation. And it turns out that the slope of this correlation is close to being -1 on average, when you average over a large population. And this has been interpreted to mean, you can translate, you can do the mapping. I'm not going to do it now. It's a simple mathematical mapping. And you can show that if this is a -1 slope, then the added volume per cycle is independent of the initial one. I crossed out the scale here, but this is initial volume, and this is added volume over the cell cycle. And this has been interpreted as some mechanistic description of division control to say that the cell is waiting to accumulate a constant added volume before it divides. Okay, and this is what the pooled ensemble gives you. Now, this is a really huge cloud, right? There's a lot of points here. And the question is, can you go back and disentangle them to the different traces, because cell division control is something that happens in every cell separately over time. It's not something that's shared between different, certainly not between different cells that live in different traps and are individual cells isolated from one another. So our question was, can we extend, and this really depends on these traces being long enough, to try to look into this cloud and separate out. So maybe there are these different traces that are parallel to each other. Maybe they have different slopes. Maybe they have both different slopes and not a single intercept. This is just a graphic representation, illustration of what we might expect might happen when we separate them to individual traces. And this is an actual data figure that shows you that although the population average trace is -1, none of the traces individually have a slope of -1, and actually every one of them has a different slope. And these are exactly the betas, the restraining forces that I talked about, that are very variable between one cell to the other. And what this means in terms of, and the fact that they all cross at a pivot point, and you can take the data and create a dynamic mapping, it means that there is some preferred cell size that everything is drawn to. There's a map, if you want to plot a mapping over phase space, then you see that individual trajectories are drawn to this preferred size, but they're drawn at different rates, because this is every bacterium is individual. And if you go back now to this, independence of the added volume on the initial volume, that only appears when you mix together and pull together different traces. When you take every trace separately, that's not a constant, actually. It deviates statistically significantly from a constant. So this reveals to us that the individual cells, they have similar control mechanisms, but there's no uniform principle of added volume that is correct for every one of them. So that that forces you to rethink these mechanisms and try to understand, what is this preferred? Why is there a preferred value for cell size? How are the different traces? How they are drawn to this, how they control their fluctuations over different time scales, etc. And yeah,
so
question.
Naama, I think this is really interesting data, actually. So do you expect that if you would be able to follow the cells for a very, very long time, theoretically, that these betas actually themselves fluctuate around some value or something, or is this really something intrinsic to those cells?
That's a great question. I don't have the plot that shows how the betas, the variance of the betas decreases, but they probably eventually oscillate around something. They probably have a range, and that range becomes smaller and smaller as the trace size becomes larger. But we were not able to we were really at the verge of statistical significance just to say that the slopes are different from one another. And you could see that the point is that where it starts to be similar was really surprisingly long. It only happens after about 100 divisions. So you might think that things are kind of mixing or becoming ergodic or mixing after a couple of generations or a dozen generations. But the time scale over which they start to become similar to one another was really much longer than we had expected. And actually, we're now in the progress of trying to figure out what affects this. And there are at least two important mechanisms. One of them is epigenetic memory. The other one is the microenvironment in which the cells live, right? And the feedback between the local medium or conditions and cell metabolism. So there are things that change locally that affect the cells. There are things that relate to epigenetic memory that affect the cells. So you really can't really answer the question about time going to infinity, because after 250 generations, more or less, the bacterium is dead. And the surprising thing is that there are remnants of this individuality. There are traces of this individuality practically for as long as the cell lives. They decrease, but they're still there.
Thank you.
Okay, so the last and really last point, it's a technical point. I don't know if you want to get into it, but what we do here, so this is cell size at the beginning of cell cycle. And again, what you can see here is the breaking of mixing of ergodicities, if you like, between two different traces. One of them is removed with respect to the other. And for this case, they retain an average distinct from one another for the entire experiment. If you take a model, the Langevin map that we constructed, and you just, with the correlations and with everything, but you draw, from one and the same global distribution, you don't see this after 250 generations. You see overlap. So time averages, if everything was behaving the same with the same parameters, then these fluctuations should be overlapping, but they're not. And so we want to quantify this, and we use what people have constructed for processes where ergodicity really breaks. For example, continuous time random walks have been studied by this group, and they've developed something that basically you can intuitively understand as follows. You look at internal variances, so each trace separate from the other has a variance, and now you average over all of them. On the other hand, you can pull everything together and compute an external variance over the entire population. Again, so this is again comparing individual traces to pooled traces. And the difference between them, properly normalized, is an ergodicity breaking measure that you can quantify how much these two ensembles are different from one another. So this measure basically is between 0 and 1. The global variance is always larger than the temporal variance of the traces. So this is always larger than 0. If temporal averages are the same as population averages, the ergodicity breaking parameter is 0, right? This is the ergodic limit. And if most of the variance comes from the fact that there are different averages, but the other fluctuations are not so important for the population, then you get a parameter which is close to 1. So these are the two extremes. And now if we take the traces that we have, the longer traces, and we look at these different parameters that we defined, the growth rate, the time, the fold growth, and we compute the ergodicity breaking parameter, we find, maybe surprisingly, that different parameters behave very different from one another. For example, the growth rate is hugely breaking ergodicity. So every trace has a different average growth rate, and that is a significant contribution to the population ergodicity, population variance. While when you pull them, you get, so this is the noise level. This is the finite size effect of the population, because otherwise, if you pull everything, then ergodicity breaking should be 0. So this gives you the baseline noise. And the physical parameters of the cell vary between being on noise level, this parameter here that I was talking about before, the exponential accumulation rate, or not rate, exponential accumulation is very close to noise level, whereas the growth rate itself and the cell size itself really differ significantly between individual cells, and that is carried over for very, very long times. And that is what we mean by ergodicity breaking. And this is work in progress, and the reason why this might again be applicable or relevant to understanding things about the cell is that it is plausible to think that some things that are more sloppily controlled can vary significantly between bacteria, but a quantity that is at the noise level and being controlled so tightly, maybe that can give us a hint about what the cell cares about when it's controlling growth and division and all these integrated processes that we're interested in. So this is a very new result, and no final conclusion as yet. Okay, so summarizing the two parts. The first part I sort of threw at you an open question that I think is really a grand question, and only partly related to the data that I showed. I think it's a really interesting question in statistical mechanics or in probability. And of course, I'm interested in it because I think it has relevance to biology, not as a purely mathematical question. And the second part, to summarize, I think taking care of these different ensembles and distinguishing between them can sometimes reveal things that are important to the cell. And although we already said this, but I'll repeat it again, although we're dealing with really phenomenological descriptions, those phenomenological descriptions and their relations can help us to understand better mechanistic issues of growth and division and related topics. So I'm putting on my collaborators again. The yeast data was done, the yeast experiments were done in collaboration with Erez Brown at the Technion. All bacterial work was done in collaboration with Hannah Salman at Pittsburgh. And some of these people collaborated with us on theoretical parts. And these are some students and postdocs. I think there are some names missing here, but anyway. Thank you. Questions now?
Thank you.
Yeah, I have a question.
Yes.
Thanks for a great talk. I was very much intrigued by this difference between the ensemble averages and these temporal averages. So some time ago, we saw international Nicola and I also looked at this question. And then we also observed that in general, for these growing and dividing cells, these temporal averages do not need to be equal to the ensemble average for a very simple reason, which is that cells that happen to grow faster, they become overrepresented in the population.
Oh, yeah, definitely.
And is this also what could be going on here, right? So especially for the growth rate, you found the strongest effect, the strongest deviation. Yes. And is that indeed simply because, you have a few cells that just happen to grow faster and they will have to grow faster for a while and they then become overrepresented in your averages?
Not only grow faster, but also divide faster. And
yeah, exactly.
Yes, absolutely, I agree. So this is something I haven't even, I think I put that up in the open questions. I mean, to tease apart these, you need more sophisticated experimental. Where are my open questions? I don't know. So maybe it was in the other presentation, but some of the open questions that I referred to was indeed to go into the tree, the entire tree of the population.
Yeah, exactly.
Yeah, which is what you're alluding to. And for that, you need, different kinds of experimental systems that enable you to follow really the whole genealogy of the population. I totally agree. Yeah.
Yeah, nice. Thank you. Yeah, maybe another question then, I don't know if other people. Maybe just to clear up my confusion, right? So in the beginning, you talked about this time scale separation. But if you have these long correlations, would you then also not expect these growth rates to be correlated from one cell cycle to the next?
Yeah. So they're correlated. The short answer is that they're correlated mainly because they have. Okay, so the universal distribution refers to the shape, right? The shape of the distribution is very robust. But as you could see from the data, the different temporal ensembles are removed from one another. There's kind of shifted. And so there's a component of the correlation across generations that stems from the average, the time average. The time averages are distinct. And they're distinct to different degrees. That's exactly what I tried to show in this Goodicity breaking parameter. So what we think is going on, and we now have a new microfluidic machine, which is really nice, that we call the sister machine rather than the mother machine, that we have two cells. This is Hannah's great work. And there's actually an interesting preprint up on the archive now with that system. But what you can do is you can follow the two sister cells and you can see them diverging from one another as a function of time. And what we find is that the growth rate is really strongly affected by the microenvironment. And that primarily affects the time average over very long time scales. On top of that, there are the fluctuations that themselves have some degree of correlation, although not that long, and they are primarily contributed from epigenetic effects. So there's kind of two different kinds of correlations that you can detect in this system.
Thank you. Nice.
So, sorry, can I ask? So, if you talk about these epigenetic effects, do you then also just mean which bunch of proteins the daughter cell inherits from its parents, like which proteome it expressed, or only how the DNA is accessible?
Oh no! This is epigenetic. Certainly, it corresponds to the level of protein. Definitely, it corresponds to the components that the mother cell is passing on. Yeah, definitely.
I have a pretty general question related to the ergodicity, a comment you made about maybe the biological significance about this ergodicity breaking. So, I think it's also in this slide, but you've alluded to the idea that maybe, the variables that more strongly break ergodicity might be what the cells care about. But I was wondering if you could motivate that intuition, why you would expect more ergodicity breaking. And
no, it's the other way around. I think if there's a parameter that the cell is really taking a lot of trouble to control and keeping fixed, then I would guess this is something that, quote unquote, the cell cares about. Because, does that answer the question?
So tighter regulation, essentially.
Yeah. I mean, it's really tricky to interpret what the cell cares about, right? It's really tricky because everything is correlated to everything else. And as far as I'm concerned, it's very hard to make out, principles. Like we said, we don't have, even if you do have molecules sometimes, and you deplete a molecule and you see that something's changing, even that is not always a mechanistic explanation for anything. And here also, but we're trying to relate these different levels with which, so first of all, it was surprising to me that there's such a big difference between different variables. Some of them are really different, some of them are hugely different, while some of them are controlled very, very tightly, numerically. So e to the phi is controlled to be 2, right? It's controlled to be very close to 2. And in some sense, you see that the cell is putting a lot of effort into doing that, right? Because the growth rates are variable, the division times are variable, but their product is somehow being kept very close to log 2, right? So that, I think, might give us a hint about how this whole interrelated control mechanism is working.
It's very interesting. And just to confirm about this delta that's also plotted here, is that the added volume?
Yeah, that's the added volume. Delta, large delta is the added volume. F is the fraction. Tau is the division time. X0 and X tau are initial and final cell sizes, and alpha is the growth rate. Yeah.
According to this view, this added phi, which is I guess the multiplicative factor, is controlled more tightly than delta.
And this, I mean, we have several different, we try to analyze this in different directions, and it always is the phi that sticks out as being the most tightly, this was a surprise to me. I don't know. It's because so the data is inconsistent with what people call a timer, right? You cannot just multiply by 2. That can't be the control mechanism because it's unstable. But still, there's something there that really keeps the multiple or the product fold change very, very close to 2.
Yeah. So could that be, so if, do the data basically argue for rather than an adder or a timer, it's a doubler mechanism?
Yeah, it can't be that either. It can't be that simple.
Sorry, but
that won't be stable either.
Yeah, that's not stable against fluctuation either. So one of the reasons, we did this exercise with the multidimensional phenotype with this model is just, this was for us an illustration how once you have dynamic coupling, everything is correlated with everything, right? And even if one variable controls things, you're getting induced correlations between other things that are just a result of the interactions. So I think that's one of the reasons why it's so difficult to figure out what is under control and what is controlling what.
Yeah, on top of this, I think there's a difference between controlling something and keeping it at the same value, like homeostasis.
Yes.
The cell might actually have a lot of variance in the growth rate because it actually is very good at growing at its optimal speed given its environment, and there's some variation in the environment. It actually regulates it very well.
Mm-hmm.
I don't know, because it could be noise and it could be like it's just on purpose, sort of.
Mm-hmm. Yeah, but you're right. That's the phenomenon. The growth rate is very sensitive to the environment, and it varies significantly between different cells.
Right.
Whereas other things are under, you want to call it homeostasis. I think probably that's a better word.
All right. So this is there's one more question. Was there another question? Just want to make sure.
I had a very quick one. Sorry.
Yeah, very last question.
Yeah. Do you think that the growth rate that's not controlling the growth rate as tightly actually provides some sort of phenotypic benefit for cells to respond quickly to the environment?
Yeah, that's probably true. And there's another thing that not to do. There's another thing that is not having to do with optimality, and that's the fact that I think the metabolic system is really sensitive to the environment, and every bacterium has, as its as implementations or as operating modes of its metabolic network, it has many different modes that it can operate on, and this can vary significantly, sensitively with sensitivity to the environment. Yeah, it could be beneficial, but it could also just be, a reflection of how rich the internal system is, and that it can use the different metabolites in many different ways. But like Peter said before, even if it's not intended for that, eventually the proportions are going to be different in the population, and it's going to be, related to maybe an advantage or a change in the representation of the subpopulations. That's certainly true.
All right, we should wrap up now. So I just want to say this was an amazing discussion. These kind of interactions have become fairly rare lately. So Naama, I really want to thank you for making this tutorial work so well, for making it be really insightful, and for provoking all these questions and discussion. So thank you so much for doing that. Just want to give you a final applause for that. Also, before switching off, I also want to thank Louise, Rutger, and Maaike for joining in as panelists. I think this was a bit of an experiment, but it was a very successful one. It made all the discussions, also with the short talks, a lot more fun and very lively. So thanks also for joining. And to the rest, hope to see you again for the next event on the 8th of December with Leonid Mirny and Agnese Seminara. See you all. Thanks. Have a nice evening.
Thank you very much. It's nice meeting you all. Bye-bye.
Thank you. Bye.